Hey guys, this is our Lab 16 review on the endocrine system. We have our exam coming up, so hope you guys are doing well. Just a quick reminder: endocrines, hormones are released into the bloodstream. So as we talk about these organs, we'll talk a little bit about how the hormone secreting cells are next to vessels. All right, and then we have our protein hormones and our steroid hormones, which go directly into the cell for their intracellular receptors. But let's get to it. This is just a reminder, we have an exam, can be a little bit overwhelming, so you got this, and, be nice to yourself. Don't know what's going on with you, but I'm certain that, you deserve to be nice to yourself. So, all right, anyways, so these are the things that we're looking at today. We have the pituitary glands. We have a nice mnemonic that's going to help us out, A sharp and B flat. We have the anterior pituitary, posterior pituitary, then we'll be looking at the pineal gland with the pinealocytes, with serotonin and melatonin. Then we'll be looking at the thyroid, which we can see here, and the parathyroid on top. Thyroid, we'll be looking at T3, T4, and then calcitonin, and then for the parathyroid, we have PTH, parathyroid hormone, which is excellent, and then we have our adrenal glands we're going to be looking at today with 1, 2, 3 layers in the adrenal cortex, and then we have our inner medulla, and so we treat these as basically two separate glands that are interrelated, the adrenal cortex and the adrenal medulla. All right, so we start off with the best one, which is the pituitary gland, aka the hypophysis. And really what we're doing is we have a nice cut through right here in the pituitary, and we're like, okay, this diagram's a little bit complicated, so what do we actually have going on here? We'll get to it in a second, but this diagram is really what we're looking at. And you can already just kind of see we have one clear section here, there's another clear section here, just going off the colors, and then there's this intermediate section right here. So we've already kind of identified the major parts of our pituitary gland, and so the question are, what are they? Well, the lecture made a point of specifying that the two major parts of the pituitary, aka the posterior pituitary, which is defined as the pars nervosa, which is right here, and then the infundibulum, which you can see right here, the stalk, is from the floor of the diencephalon. It's actually nervous tissue, and we'll see that. And then you have the same tissue that came from like the roof of the mouth. Epithelial tissue grew up, met the floor of the diencephalon, and they sort of merged together to form the pituitary gland. And as a result, you get two very distinct sections: the anterior pituitary on this side and the posterior pituitary on that side. All right, and so we have the anterior pituitary, with the major part of it is the pars distalis, which we'll talk quite a bit about with the A sharp and B flat. Then you have the pars tuberalis, which you can see wraps around the infundibulum, which is part of the posterior pituitary. And then you have the pars intermedium, which is right here. And so we can start to get a sense. All right, so this is anterior pituitary, this is posterior pituitary, and this is the pars intermedia, this is the pars distalis, and this is the pars nervosa. Looks like nervous tissue, and in fact it is. So again, same thing. We have one section right here. This is pars distalis of the anterior pituitary, which is also called the adenohypophysis. Hypophysis stands for pituitary. Adeno is anterior. Perfect. So there we have that, and then we have our posterior pituitary, and again, it's kind of washed out looking. It is our nervous tissue, and this is the pars nervosa right here, okay? And then in between, we have our pars intermedium, and we'll talk more about that in just a minute, okay? So here we have it. One more time, we like repetition, so pars nervosa, but we're becoming champions at this. Here we have pars intermedium, which is part of the anterior pituitary again, and then pars distalis, also part of the anterior pituitary. And then again, what's our third part of the anterior pituitary? It is the pars tuberalis, which wraps around the infundibulum, which is part of our posterior pituitary. Okay, a little bit of repetition. So now we're in the fun part. This one is the pars distalis, which is right here. This was this section right here, which is more basophilically staining. And here we have two main cell types. So we have the acidophils, which are eosinophilic, relatively speaking, and we have the basophils, which are basophilic. So if we just look at this section, you can see that we have cells that are lightly staining, and then we have cells that are darkly staining. And so our lightly staining cells are acidophils, and our mnemonic for acidophils is A sharp, and A is for acidophil, S is for somatotropin, aka growth hormone. It's produced by somatotropes. And then P is for prolactin, produced by mammotropes. And both those cell types reside in here. They are the acidophils. Well, we have to determine if this acidophil, these acidophils right here, if they're of the variety that produces somatotropin, aka the somatotropes, or the prolactin producing types, the mammotrophs. No, all you have to do is recognize that these are in fact acidophils because they are lightly staining. And you remember acidophil, A sharp. All right, then we have our basophils, and our basophils are the opposite. They're darkly staining. And for basophils, we have B flat. So A sharp, B flat, B for baso, and then F for follicle stimulating hormones from our gonadotropes, L for luteinizing hormone from our gonadotropes, A is for ACTH, which is adrenocorticotropic hormone from our corticotropes, and then T from our thyrotropes for thyroid stimulating hormone. Okay, so we have our basophils with B flat, our acidophils for A sharp, and it's S and P. Okay, there is a third type of cell here, and we can see some occasionally. So they're kind of these washed out looking cells. Here's one right here. Here's another one right there, another one right there, one right there. You can see them around. And so they are inactive or depleted versions of basophils or acidophils that have released their contents, right? And so these hormones are produced in the acidophils and basophils and give the acidophils and the basophils their color. But when they release that hormone, then they will be chromophobes, washed out, or looking washed out overall. All right, final thing is we have our fenestrated capillaries, and they're fenestrated because the hormones need to be able to go into the blood. And so how would you find this? Well, look for the red blood cells. So you can see like all these guys right here, red blood cells. If you zoom in, you can really just see the edges of them, but you can see red blood cells here. And so that is some sort of fenestrated capillary. All right, so moving on, we have the pars intermedia right here. Pars intermedia is nice and easy because you have pars nervosa and then pars distalis, and it's the part in between that has these colloid-filled crypts, which are the remnants of Rathke's pouch, which is really the hollow that was right here from the epithelial tissue from the roof of the mouth during development. And it's going to be filled with so colloid-filled cysts, and so you see those, you're like, okay, I'm in the pars intermedia. But the major cell type that's in the pars intermedia is actually one of the cell types that's in the pars distalis, and that is our basophils. So our pars intermedia actually has basophils, and we know that that is B flat. Okay, and so that's just another piece of thing, hopefully, to remember that pars intermedia is actually part of the adenohypophysis, aka the anterior pituitary. Hypophysis is pituitary gland. All right, moving on, we have the pars nervosa. Pars nervosa is right here. And the pars nervosa is interesting because the cells in the pars nervosa are not going to be producing the hormones that they release into the bloodstream. In fact, it's actually axons from the paraventricular nucleus of the hypothalamus and the supraoptic nucleus of the hypothalamus that produce the hormones, and then they send those hormones down into the pars nervosa of the posterior pituitary, where they're then released into the blood. And so the pars nervosa is a neurosecretory site for neurons whose cell bodies are in these two nuclei in the hypothalamus. They're poorly stained. It has like a frothy appearance. Doesn't this just kind of look like, nervous tissue to you? It does to me, right? Sort of frothy looking. And why is that the case? Well, it's a lot of axons, but then you have all these little cell bodies, and these cell bodies are called pituocytes, which are supporting cells. All right, they're not involved in hormone secretion. They're supporting cells, and then we sometimes have these brown stains. This is lipofuscin. It's like a brown waste pigment from long-lived cells. And then the question is, okay, well, what are the hormones that the paraventricular nucleus and the supraoptic nucleus, what are they releasing? Well, oxytocin and vasopressin. Oxytocin causes uterine contraction, stimulates milk production. It targets uterine smooth muscle and mammillary myoepithelial cells. Vasopressin increases water absorption in the kidneys, and it targets the collecting duct in the kidneys. It's also a vasoconstrictor. Okay? All right, so oxytocin and vasopressin actually coming from the hypothalamus, but getting released in the pars novosa of the posterior pituitary. So how does it do that? That is a great question, and it relies on these things called Herring bodies. And so Herring bodies are elongated, homogeneous, eosinophilic structures that look like a small tuft of cotton candy or cotton swab. And so you're like, okay, well, what about all these little cells? Remember, those are the pituocytes, which are supporting cells for these axons that are coming down from the hypothalamus. So here would be a pituocyte, and then here's the cell body that's in the hypothalamus. It's sending its long axon down, and it's sending those antidiuretic hormone or oxytocin hormones in these axons with these little bulges that are called the Herring bodies, okay? And then so how do you go about find them? Well, they look like kind of cotton swabs, very washed out. You can see here, here, here. Here's a really good one. Here's another good one. But all of these cells, again, are the supporting cells, the pituocytes. These guys right here are the ones that are containing your antidiuretic hormone, aka vasopressin, and your oxytocin. Okay, so Herring bodies, and then our pituocytes, and that is our pars novosa. And our pars novosa is the dumping ground for paraventricular nucleus and supraoptic nucleus. All right, so supraoptic. And they both release both of those hormones, but let me just double check that. Yep, so they both release oxytocin and vasopressin, also called ADH, antidiuretic hormone. All right, so just a review of the pituitary system. So we had our cut right here. We had our anterior pituitary, which had three parts: the pars distalis, the pars intermedia, and the pars tuberalis. In particular, pars distalis, we said that there was A sharp and B flat. B for basophil, and then flat for follicular stimulating hormone, luteinizing, ACTH, and TSH. And then A sharp for growth hormone, aka somatotropin, and then P in the sharp for prolactin. All right, blood supply, we had the, this is anterior pituitary, we have the superior hypophyseal artery, and so you can have like thyroid releasing hormone from the hypothalamus will come down, go into this artery here, which will go into the pars distalis, and then cause the basophils of the pars distalis to release thyroid stimulating hormone. And then on this side, you have the inferior hypophyseal artery, such that when the axons from the paraventricular and supraoptic nucleus go down, they can cause, when they release oxytocin and antidiuretic hormone, they can go into the inferior hypophyseal artery and into the bloodstream. All right, so not so bad. Next up is the pineal gland, and you can see that there. And the idea is that the pineal gland releases two hormones. It is serotonin and melatonin, which both have to do with regulating sleep. And basically, just that, we can see it here. The pineal gland is relatively straightforward. There's not too much you need to know. We have glial cells acting as supporting cells, and then we have our pinealocytes, which are secreting the melatonin and serotonin. They are the bigger cells, and then the glial cells are the smaller cells. But pineal glands are easy to identify because they have this stuff called brain sands, which is calcified concentrations of carrier proteins, also called corpora arenacea, which I probably mispronounced. All right, so more on the pineal gland. Stroma, the pineal gland is covered by a capsule layer of connective tissue. It's continuous with the pia mater overall, which we know covers the brain as the innermost layer. connective tissue layer. Then we have the functional cells. As we already said, we have the pinealocytes and then the glial cells. And you can see that they form tiny little islands that are surrounded by loose connective tissue. We would call these islands cords. So these islands are called cords, and then they're surrounded by connective tissue overall. And so it looks like dense irregular connective tissue. And you can see the brain sand here. In terms of, what is what, the larger ones are the pinealocytes, the smaller ones are the glial cells. The small guys are glial cells, but most of the cells here are pinealocytes. They're bigger overall and more numerous. All right, so we have the islands, which you can see here. They're called cords. They have the pinealocytes and the glial cells. You see that we have connective tissue around these cords, and then we have the brain sand, corpora arsenacea, and then we do have some blood vessels around here. A little bit harder to pick up. You'll have to look for sort of the eosinophilic RBCs, red blood cells. All right, so more on the pineal gland. This is the exact same thing as we said. Pinealocytes have sort of prominent nucleoli, right? You can see it kind of looks a little bit lacy, and they produce melatonin and serotonin. Lots of connective tissue, and our glial cells are much smaller. You don't have to distinguish them in our class. All right, then we have the thyroid glands. And so we are very familiar with the thyroid gland from clinical skills and anatomy, but in particular, we see that on the front side, we have the thyroid. On the back side, kind of embedded in the thyroid, we have the parathyroid. So we'll talk about them both. But the thyroid is going to give us thyroid hormone in the form of T3 and then T4. T3 being the active component. And then we have calcitonin produced by the parafollicular cells. And then for the parathyroid glands, we have parathyroid hormone. All right, so basic setup of the thyroid gland is all about follicles. So we have basically follicle cells, which are going to produce T3 and T4, and they're going to be stored in colloid in the lumen of these cells. So this is all colloid. And the idea is that they are going to, under the influence of thyroid stimulating hormone, they will become bigger and more active, but when they're less active, they tend to be a little bit smaller. And so these ones look fairly big, fairly active. Maybe we can find an example of some squished ones. All right, well, not so much on the slide, but these are our follicular cells, and we have the colloid. And then outside of that, we have our parathyroid cells, which can sometimes be embedded in the basement membrane of the follicular cells, but they're generally oriented in clusters outside of the follicles. And so they kind of look for them, but here's one right here. These appear to be some, and they're going to produce calcitonin, which lowers blood calcium. Okay, so yeah, here's an example. Calcitonin cells, they're independent of the function of thyroid follicles, and they store granules of calcitonin intracellularly. All right, so more on this. Basically, structure of the thyroid is we have two main lobes, right? We have the right lobe, we have the left lobe, and then sometimes you have this little pyramidal lobe that extends upward. The center part, though, is the isthmus. And thyroid, you can see, is made up of, as we said, all of these little follicles. And the thyroid gland is separated into lobules by connective tissue. Here we see that there is a thin layer of connective tissue over the thyroid overall. And then the parenchyma again is like, what are the functional cells? We have our follicles. All right, so again, follicles, simple cuboidal epithelium. That's what we saw here. Here's a better example of, we said that they get larger upon stimulation from thyroid stimulating hormone. So notice how they're like pretty flat over here. Here they might be a little bit chunkier. So the bigger they are, they get bigger with stimulation and therefore more active. All right, then we have, and so what do they produce? So shorter in height when inactive, taller when active. They produce thyroxine, which is T4, the more stable version of thyroid hormone, which will be converted into T3 throughout the body when it needs to be used. Parathyroid cells are also called C cells. C is for calcitonin. And as we said, they're located in the in between the follicles, sometimes in the basal lamina of the follicle. they're large cells that stain poorly overall, right? So right here's one, here appears to be another one, potentially. Okay, and then what does calcitonin do, right? Well, tone down calcium. It's going to say lower calcium levels, tone down calcium levels by depressing osteoclast activity, and then increase excretion of renal and intestinal excretion of calcium, right? Lower calcium, tone down calcium however you can in the blood, pee it out, poop it out, stop pumping it into the blood, aka lower your osteoclast activity. All right, capillaries in the thyroid are pretty cool. We're given this electron microscopy picture, and so the idea is that the network of capillaries, they're fenestrated one, but they surround each follicle. It looks like a basket, right? So you can see that this looks like a basket. Inside of this, there would be a follicle, right? But these are all capillaries and fenestrated to pick up the hormones from the thyroid, right? T3, T4, and then calcitonin. All right, and then in terms of figuring that out, you got to look for red blood cells. So here's an example here. Here's maybe some red blood cells overall. All right, here's our parafollicular cell. But yeah, okay. This is just an example. We don't necessarily need it, but this is an example of hypothyroidism. So Graves' disease, this is a normal thyroid, right? Notice how we have these huge, nice lumens full of colloid, just stored, ready to be released. And Graves' disease is where you have autoantibodies that are binding to the thyroid stimulating hormone receptors and causing a bunch of T3 and T4 to be released. So notice if this guy's full, this is what they look like when they're empty, right? Here's where they've been active. So you got like, I believe this is air bubbles. And so basically you can see that the follicles are relatively empty, which means that you have overstimulation of T3, T4. So this would be Graves' disease. All right, then you have hypothyroidism, and in this case you have Hashimoto's. The follicles are being destroyed. You can see we have very tiny follicles overall, and it's just autoimmune destruction of the follicular cells, so less T3, T4 being produced, lower metabolic rate, you're going to have weight gain. All right, then you have parathyroid hormone, which we said is on the backside of the thyroid gland. We have two cell types. You have chief principal cells, which produce parathyroid hormone. And you're like, what does parathyroid hormone do? It's the opposite of calcitonin. So you think, as in not cool, right? Not cool, PTH, not cool. Versus calcitonin, calcitonin is really cool, it tells things to calm down. PTH, not cool, it tells things to crank it up, right? So PCTH, increase osteoclast activity, liberate calcium from the bone, increases reabsorption of calcium. You're going to raise calcium in the blood. All right, and then we have oxyphils overall, which we're not responsible for the function of those, and we weren't told what they do. All right, but notice that we have adipose cells in the parathyroid glands. These big fat red cells are the oxyphil cells. Then most the other cells that are smaller, basophilic, they are the chief cells, principal cells. All right, so here's just more of that. Stroma, we have a capsule just like before, but in this case we also have the capsule comes down and forms trabeculae to separate our thyroid into sort of incomplete lobules. And so there is a decent amount of adipose tissue overall. You'll notice that we have clumps right here, and the red ones are the oxyphils. And again, what do? We don't need to know. And then the purple basophilic ones are our principal cells, which release PTH. Okay, so here, red ones are oxyphils, contain a lot of mitochondria. It increases with age, the number of them do. And then principal cells, again, the opposite of calcitonin, relatively small, very numerous. All right, then there was a question of parathyroid versus anterior pituitary. They look a little bit similar, right? Anterior pituitary was B flat, A sharp, right? And so here we have, our oxyphils and our principal cells. And here we have our basophils and our cytophils overall, and then we have our chromophobes. But notice how, the basophilic population is huge. There's tons of little tiny ones. This should be pointing to right here. And in comparison, the anterior pituitary doesn't have quite so many of these basophilic cells, right? So it's a good sense it's parathyroid, and then the presence of adipose tissue is not there in the anterior pituitary. All right, final one is the maybe second best one, adrenal glands, because it's really two separate organs. You have the adrenal cortex and the adrenal medulla. Medulla is on the inside, cortex is on the outside. And so the adrenal cortex produces a bunch of steroid hormones, and the adrenal medulla is doing protein hormones. All right, but overall you have a connective tissue capsule, and that connective tissue will extend downwards into the cortex as trabeculae. The cortex itself has three regions. You have the zona glomerulosa at the very top. We have the zona fasciculata, and then the zona reticularis, and then the adrenal medulla, which we'll go over more in detail. Starting with the first layer of the adrenal gland, the adrenal cortex, excuse me, the zona glomerulosa. And glomerulosa is meant to be ball in Latin. And so you look at it and you have all these tiny little balls. And so that's, your hint that this is in fact, plus you're on the outside, it's the most apical layer of the adrenal cortex. This is the zona glomerulosa. What does it produce? It produces aldosterone, which is a mineral corticoid. Corticoid is just saying it's coming from the adrenal cortex, and then the first word is what it regulates, so minerals. In particular, it produces aldosterone, and what does aldosterone do? It causes reabsorption of sodium in the kidneys, and it is regulated in particular by angiotensin 2 overall, and a little bit by ACTH from the pituitary gland, but primarily angiotensin 2 from the RAS system. Okay, so zona glomerulosa, think aldosterone, mineral corticoids. Okay, mineral corticoids, zona glomerulosa. You got balls on the top. Okay, then you have the zona fasciculata, and that's next. Zona fasciculata. Fasciculata is bundles, so you have a bunch of little bundles. And overall, you can see more of those. Here's a nice cross section. You have these foamy cells, and it produces glucocorticoids. So corticoid, just a hormone released from the adrenal cortex. Gluco means it deals with glucose levels. So cortisol is, or stress hormone, right? It's going to promote metabolism and basically upregulate the amount of glucose available for usage. So add glucose to the blood. All right, so cortisol, glucocorticoids. So you went from glomerulosa with aldosterone, mineral corticoids, to glucocorticoids. Okay, and then this is dependent on ACTH from the anterior pituitary. All right, next you have the zona reticularis. So again, zona glomerulosa, then you have fasciculata, and then you have reticularis. Reticularis is two-toned generally, and it's right here. You can see it here, and it's going to produce gonadocorticoids, in particular dihydroepialdosterone. Okay, so how can you remember that this one is gonadocorticoids? Well, terrible way to think about this perhaps, but gonadocorticoids, testicles are towards the bottom of the body, right? And so the lowest level of the adrenal cortex, zona reticularis, produces gonadocorticoids, dihydroepialdosterone, and it's stimulated by ACTH, just like the zona fasciculata. All right, so not too much there, two-toned overall. Then you have the medulla, and so you can see that here. This is this whole inner layer overall, and the medulla is going to produce epinephrine and neuroepinephrine, mostly epinephrine. The cells that do this contain an enzyme called phenylethylamine N-methyltransferase, PNMT, which converts neuroepinephrine into epinephrine. Okay, PNMT is induced by glucocorticoids from the zona fasciculata. Okay, and then what are the cells that are in here? Well, we have chromaffin cells, which are modified postganglionic neurons, sympathetic postganglionic neurons from the neural crest, and they secrete epinephrine, neuroepinephrine from nervous system input, right? So Here you can just see an example: preganglionic sympathetic neuron synapses on the chromaffin cell. Chromaffin cell releases epinephrine and neuroepinephrine, and they contain PNMT, which is stimulated to be released by cortisol from the zona fasciculata. Okay, so not too bad. Neuroepinephrine, epinephrine. All right, blood supply of the adrenal cortex is the very last thing to go over. So we have arterial supply is limited to the cortex, and then blood will go from the cortex into the medulla, where it will be drained by the medulla into veins. But you don't have veins in the cortex, you have veins in the medulla. So you start with your capillary arteries on the top. It's located in the connective tissue. They bring blood to the gland, and they give rise to cortical medullary arterioles. And so you have your cortical arterioles, which break up and become the subscapular plexus and the sinusoidal vessels. And you also have medullary arterioles, which go all the way down into the medulla before they branch. All right, so the cortical arterioles branch immediately into the subscapular plexus and sinusoidal vesicles, and then the medullary arterioles go directly into the medullary plexus in the medullary. Okay, but basically, yeah, okay. Then we have, so more on this. Basically, here's just an example. We have our capillary artery coming in. It gives rise to the cortical arteriole, which you can see branches and supplies the adrenal cortex. Then you have the medullary arteriole, which goes all the way down without branching into the medulla. But you do have drainage. Basically, these do anastomose eventually within the medulla, as in the medullary plexus. So the cortical capillaries, these guys, will drain into the medullary plexus. And this is important because we said that the neuroepinephrine and epinephrine released by the medulla does so under the influence of glucocorticoids, aka cortisol, from the they can be stimulated, or excuse me, PNMT production is stimulated by cortisol from the zona fasciculata. All right, and this is possible because the cortical capillaries drain into the medullary plexus. Okay, and then the medulla has veins, whereas, that's the ultimate transport out of there. All right, so this is just that, no veins in the adrenal cortex, so it has to drain into, everything has to drain into the medulla eventually. And yeah, so here we have, these are right here, the cortical capillaries. These are medullary, that's the medullary plexus right here, and this is just our medullary vein. All right, so quick review. Here we have our pituitary gland. This is pars nervosa, pars intermedia, pars distalis. Great. Then we had, what produces what? Well, we had B flat, A sharp. We'll let you guys look at that. Here's a transmission electron micrograph of pars distalis in particular. This is a acidophil in particular. Of the acidophils, this is a somatotrope, which we know is also producing somatotrope. Otropin is also a growth factor, growth hormone, and they are these nice circular little granules overall. All right, more on the pars nervosa. Remember, pars nervosa is made up of axons, and you can see the herring bodies, right? This is a herring body, and all of these guys are the pituocytes, which are supporting cells for the herring body, the axons that have the herring bodies. All right, thyroid glands, we have our cuboidal follicular cells. The colloid contains iodinated thyroglobulin. You have T3 and T4 produced by these follicular cells, and then they get bigger when they're stimulated by a thyroid-stimulating hormone. Then we had our parathyroid cells, which are large and kind of washed out, or outside of the follicles. And here we have some TEMs. This is a parafollicular cell right here, and again, parafollicular cells are producing calcitonin, and so we have these little tiny granules of calcitonin. And then you have colloid here. This is a follicular cell, nice and little, and then a fenestrated capillary. And what does calcitonin do? Tones down calcium in the blood, so depresses osteoclast activity, decreases calcium liberation from bone, but increases output of calcium via peeing and pooping. All right, then we had the adrenal gland. We just went over this, but so we have zona glomerulosa, zona fasciculata, right, the bundles. Then you have the zona reticularis, which produces the gonatocorticoids. Okay, and then adrenal cortex. Here we have medulla, the chromuffin cells, and then here we have zona reticularis. Here's that in an electron micrograph, and you can see here, this is the medulla. And so you may recall that I said that the layers, these guys all produce steroid hormones, which are, cholesterol lipid based. And so they aren't going to have any granules because as soon as those are made, they can diffuse through the membranes. But the medulla, which is epinephrine and norepinephrine, which are protein based, and they're going to be stored in granules. And you can kind of see that here a little bit. Yep. Okay, and I think that's about it. Here, zona fasciculata, large number of lipid droplets, and yeah. Okay, and here is anterior pituitary, and they are producing, you have A sharp and B flat in the anterior pituitary. In this case, this is somatotrope, making somatotrophin, aka growth factor, so that is an acidophil. And you have large circular granules. This is the only acidophil that we've seen an electron micrograph of. All right, thyroid gland, their fenestrated capillaries, and we already saw this picture. Okay, and then here's parafollicular cell, aka C cell, are calcitonin cells, and they have many small granules, and then you have large lysosomes, and yeah, okay. All right, and I think we went over this one. Yep, and we went over that one, and that's it. All right, well, that was really long, but good job, guys. And, as we said, you got this. Be kind to yourself. Here's the summary, but you can do it. All right, bye, guys.